This is my presentation on biotechnology. Aspergillus oryzae is a filamentous fungus with septate hyphae and is in the division Ascomycota. It is a pathogen and allergen in humans and animals. The understanding of the fungus Aspergillus oryzae can be used in the biotechnology industry. They are identified by tiny spores. The growth conditions for Aspergillus oryzae is a temperature of between 24 and 26 degrees Celsius in aerobic conditions. Conclusions from statistical analysis. From the results of the experiment, it was clear that certain concentrations of glucose affected the rate of growth of the Aspergillus oryzae, as increasing the concentration increased the number of colonies that grew on the agar plate. Therefore, as the concentration of glucose agar increased, the amount of Aspergillus oryzae grown also increased. From the results obtained, I discovered that 1% glucose gave a small amount of growth, followed by 2%, 4% and 5%, with 5% giving the largest amount of growth. Therefore, the best concentration to use is 5% when making up the medium to allow optimum growth of the cultures. The results also showed that the antibiotic tetracycline is the most effective against Aspergillus oryzae, as it had produced the largest area of inhibition. From the results, I also discovered that the antibiotic psilocybin and sulfafurazole are the least effective against Aspergillus oryzae, as they produce the smallest area of inhibition. This means that the tetracycline antibiotic had a greater effect in stopping the growth of Aspergillus oryzae compared to the other samples. Growth tests are used to determine the nutrients required for growth. This can be worked out by growing the organism on different mediums until there is a maximum growth. Optimum temperature. There are different optimum temperatures for different microorganisms. Optimum pH. Different microorganisms have different optimum pHs. The oxygen requirement. Some microorganisms need oxygen and some don't. These tests can help determine all of the conditions for a specific microorganism so that it can be applied in the biotechnology industry. Methods for controlling the growth of microorganisms. Temperature. Low temperatures can reduce enzyme activity and slow the growth of microorganisms. Additionally, most microbes cannot survive at high temperatures. Therefore, heat can be used to sterilize surgical instruments or equipment used in food manufacture. Pasteurization. This method is used for milk and beer. When a temperature of 72 degrees Celsius is applied for 15 to 20 seconds, the number of pathogens are reduced so that they cannot cause diseases. Water potential. Placing food in strong salt or sugar solutions preserves it because water leaves bacteria cells by osmosis, and the cells cannot carry out their metabolic reactions. Antiseptics. These can be used to kill microorganisms on living tissues, such as skin. What is biotechnology? Biotechnology is the use of biological systems found in organisms or the use of the living organisms themselves to make technological advances and adapt those technologies to various different fields. Modern biotechnology provides breakthrough products and technologies to combat rare diseases, reduce environmental footprint, feed the hungry, use less and cleaner energy, and have safer, cleaner and more efficient industrial manufacturing processes. Biotechnology is used in transgenics. This is where a gene is taken from one species and implanted in another in order to introduce a desirable trait. For example, a transgenic animal is one whose genome has been altered by the transfer of a gene from another species. Phylogenics is represented by a tree diagram called the phylogenetic tree. It shows the relationship between groups of organisms, particularly the differences and similarities among them. This allows scientists to investigate the phylogeny of disease organisms in order to determine their rate of evolution. Bioinformatics involves the use of computers to collect, organise and use biological information to analyse biological data such as evolution. Genetic modification is a set of technology used to change the genetic makeup of cells, including the transfer of genes within species to produce improved organisms. For example, corn has experienced some of the most dramatic genetic changes over the past few thousand years. The crop was derived from a plant known as teosinte, a wild grass with tiny ears that bore only a few kernels. Over time, farmers selectively bred the teosinte grasses to create corn with large ears bursting with kernels. How is Aspergillus oryzae applied in the biotechnology industry? The biotechnology industry had made use of the enzymes of Aspergillus oryzae for the food industry. It's safe to use in food production because it doesn't produce carcinogens. It is one of the most powerful secretory producers of protein among the filamentous fungi and has been used for hundreds of years in Japanese traditional fermentation industries, including Oriental alcohol beverages such as sake, which is a Japanese rice wine. The fermentation of genetically modified Aspergillus oryzae is a major source of recombinant chamogen, which is used to curdle milk to make hard cheeses. Amylase of Aspergillus oryzae are used in the food industry for baking, brewing, production of cakes, fruit juices, and starch syrups. It can be added to the dough of bread to reduce the starch in the flour into smaller dextrins, which are then fermented by the yeast. The addition of alpha amylase to the dough results in enhancing the rate of fermentation and the reduction of the viscosity of the dough, resulting in improvements in the volume and texture of the product. Viruses and bacteria can also be used in the biotechnology industry. Viruses can be used in medicine as they can be used as vectors or carriers that take the required material for treatment of a disease to various target cells. They have been studied in management of inherited diseases and genetic engineering as well as cancers. They can also be used in agriculture. The modification and genetic engineering methods can be used to make modified genomes that can be carried into plants and animals by viruses acting as vectors. This method can lead to more productive transgenic animals and plants. In the food industry, lactic acid bacteria such as Streptococcus are used in the manufacture of dairy products such as cottage cheese, cream cheese, sour cream, buttermilk, and yogurt. In the pharmaceutical industry, bacteria are used to produce antibiotics, vaccines, and medically useful enzymes. Most antibiotics are made by bacteria that live in the soil. The biotechnology industry uses bacterial cells for the production of biological substances that are useful to humans, including fuels, foods, medicines, hormones, enzymes, proteins, and nucleic acids. Genetically modified Aspergillus oryzae is used for the large-scale production of lipases. Lipases are biological enzymes which digest fat, and so are used in biological washing powders. The use of enzymes in detergent formulations enhances the detergent's ability to remove tough stains and making the detergent environmentally safe. Fermentation. Fermentation is a metabolic process that produces chemical changes in organic substrates through the action of enzymes. Microorganisms are grown in a bioreactor in which the microorganisms go through the log, lag, stationary, and death phases. Using these bioreactors in the industry allows the scientists to control and monitor the growth of organisms. There are two types of fermentation, batch and continuous. It is necessary that the microorganism is supplied with the optimum temperatures and conditions for growth such as the correct nutrients, optimum pH, and optimum temperature. The time allowed for the microorganism to ferment is critical, as if not enough time is allocated, the desired processes will not have time to occur. But if too much time is given, overpopulation of the sample can occur and suffocate the organism, decreasing the amount of product being created. By growing Aspergillus oryzae in different environmental conditions and mediums, it is possible to discover the optimum growing conditions for the fungi so that biological reactions can occur. This makes it possible for large quantities of the microorganism to be grown effectively and efficiently. The byproducts of the process can be isolated and used in the biotechnology industry.